Okay. Good evening. Learning gives creativity. Creativity leads to thinking. Thinking provides knowledge, and knowledge makes you great. These are the words of our former president, Dr. APJ Abdul Kalam. Good evening, everyone. On behalf of IMA MSN Kerala, I would like to welcome all of you to the third session of the much awaited National Scholarship Examination, Enigma 2021, for all our first year medicos. Here we have Dr. Varsha ma'am, who is a national faculty in biochemistry, with us to handle our session on metabolism of carbohydrates. Welcome to the session, ma'am. Schedule of the program is from May 18 to 24, which includes interactive sessions, workshops, and finally, the much-awaited scholarship exam. We also provide a 7-day free trial access via eGurubal app for all the registered participants to augment their journey towards the endpoint. Handing over the session to you, ma'am.
Thank you, Fiba. Thank you very much. Let me introduce myself. Dr. Varsha Gupta, and I have done my PhD and MS from University of the Sciences in Philadelphia. And I am teaching biochemistry since almost 17 years. And earlier I was a chemistry student, and I really don't know how I became a lover of biochemistry. It's beautiful. And in India, to my surprise, since I'm here in India since 2 years, that everybody undermined the biochemistry subject, which should not be the case, because biochemistry is a heart of everything being you as a medicos learn. Everything will revolve around the biochemistry, and you take any subject, you will have a biochemistry touch to it. So it is very important, really very, very important, because back in States, my pre-med students really had hard time in biochemistry, and that is the biggest portion of their entire course to get entry into the medical school. And even in the medical school, biochemistry is given the priority. So just to squeeze everything in, or you can say to summarize everything, biochemistry is good. And I love biochemistry because it's a chemistry behind the biology, which is your subject. The chemistry behind your pharmacognosy, you can say your pharmaceutics, you can say your pharmacology, anatomy, everything has a chemistry to it. And that is the biochemistry. So today I'll be taking metabolism of carbohydrates. So if I take step backwards, carbohydrates is something that we eat in our food. And when we have any food material that we eat that is composed of the carbohydrate, that has the amino acids in the form of the proteins, that has the fat, like you have all the mix up of the carbohydrates, proteins, and fats in your food. That is the first step when you have food in the mouth and you chew it. Then based upon various metabolism, carbohydrates are broken down into the glucose units that are a monomer, or you can the simplest unit. Proteins are digested into amino acid that you study under the protein metabolism, and the fats are digested to fatty acid that you study under the fat metabolism. So our major concept for today is the carbohydrate metabolism, and I will try to give you the glimpse how really we teach and what should be the best way to teach or read anything so that you have the keywords. For me, keywords is the best way to remember everything in the exam, be it any subject in the whole wide world, right? So carbohydrates are chewed into the glucose. That is a step 2. And after step 2, you have the stage 2 or step 3, in which pyruvate is getting converted into ultimately into your FADH2, NADH, your ATPs, and that is what we are looking for. We are looking for the energy, right? That's why we eat food. Every entire system in your body that is functioning needs the energy, and the energy currency, I call it an energy currency, is the ATP. Like Indian currency is INR or rupees. US is the dollars. So energy, the currency of your body is in the form of ATP. You should have the most needed amount of ATP in your body. That if the ATP level goes down, you feel hungry. And that's why you want to eat food consume more food so that you replenish the ATP stores in the body. That comes via all these pathway. First step, the complex ones, the carbs are broken down into the smaller units, that is the glucose, which is then taken to the second stage, that is a pyruvate, which pyruvate from the carbs, amino acids from the proteins, and the fatty acids from the fat. They all combine together to one pathway. That is, you can say all the rivers. Can you imagine how it finally goes and merge into the ocean? Similarly, all these three pathways finally go and merge into the citric acid cycle in the form of the acetyl coenzyme A. That is a very interesting pathway, TCA cycle, because it is a cycle. And whenever there is a cycle, it means there is a catalyst involved in it. So that is very interesting and funny part, the TCA cycle. From the TCA cycle, every carbon is exhausted, and then you are left with the FADH and NADH, that is the electrons. And from that electrons, your oxidative phosphorylation and electron transport chain finally convert those electron gradient into the proton gradient, and finally you have this magic wand ADP synthase, which synthesize the ATP from the electrons and the proton gradient. And that is amazing, really truly amazing to see that. So today in my lecture, I'll be just touching a little bit of the carbohydrates, how specifically the glycolysis I will touch, and then I have something for you in house, that what is the new medication that is coming in the market, that is for COVID, SARS-CoV-2, COVID-19, and that is related to the glucose, how glucose thing can be used to treat such deadly disease. So first of all, about digestion of carbohydrates, we all know that the majority of the dietary carbohydrates are starch, sucrose, lactose, right? And this is the entire diagram in which I'm trying to squeeze in. You have the food, starch, lactose, and sucrose generally in like major. Oh, I'm sorry. That the part I have missed. My apologies for that. Portion I was talking about, that today we will be discussing about the digestion of the carbs and then the glucose transporters and the few key points of the glycolysis and so on and so forth. So the majority of the dietary carbohydrates are in the form of the starch, sucrose, and the lactose, right? And this is the diagram. I picked it from a book, very known book, and this is showing you this cartoon that you eat the food. And the majority of food is comprised of the lactose, sucrose, starch, right? And in your mouth, you have the salivary amylase, which chops this food down. First, you macerate the food using the teeth, and then the salivary amylase is an enzyme which chops it down into the complex carbs, into a little bit simpler carbs. And then you have the sucrose, lactose in the stomach. Now, there are three monosaccharides, I call it, the glucose. If you combine that glucose with another glucose, then that forms the maltose. For those who don't know it, I'm just giving you, refreshing it to you. Then you have another important monosaccharide unit, fructose, which combines with glucose to gives you the sucrose. And that there is third one, that is the lactose, which is composed of glucose plus galactose. Okay. So now coming back to this point, in the stomach, now the salivary amylase had digested your complex carbohydrates into the simpler ones. You have the sucrose and lactose in the stomach, which further by alpha amylase are broken down into smaller portions. And finally, you have these monosaccharides. I'm not going to go in detail in this rapid review, I call it. Okay, so you have the glucose, you have the fructose, you have the galactose. All three monosaccharides are there now in the intestine, and then that will be taken down further to digest. Finally, like you can say, the chutney in the form of a chutney, then you extract every single carbon from the glucose, your lactose, and then your galactose. That is a 6-carbon thingy thing. And how you do it, that we will do for glucose today, right? So digestion of carbohydrates in the intestine, I told you, first of all, was done by the pancreatic enzymes, right? The pancreas secretes the alpha amylase enzyme, very, very important enzyme. In the enzyme chapter, we really talk about alpha amylase enzyme. It is a diagnostic marker also for the several diseases, okay? Now, the products of the digestion, after digestion by alpha amylase, you have the maltase and the isomaltase, right? They are composed of 2 glucose units. Then in the intestinal cells, now you have the few enzymes that are located in their brush border. Glucoamylase, isomaltase that digests the isomaltase. Sucrase converts the sucrose into glucose and the fructose. These are the monosaccharides, and this sucrase is an enzyme. Whenever you have a suffix of ase, that is an enzyme, okay? Lactase digests the lactose into glucose and the galactose, right? So you have now glucose. We are talking about the glucose in this chapter, in this session today. So glucose is there now in the intestine, and then it should be taken down inside the cell to further degrade it. That is done by the transporters, and you have like various glucose transporter, basically 15, but 4 are the major ones: GLUT1, GLUT2, GLUT3, GLUT4, and sometimes the GLUT5, right? So these are the glucose transporters for uptake of glucose from the blood inside the cell. So these are majorly 4 kind of blood I want to talk about, and usually asked in most of the examination. I'm talking about the topmost is the USMLE, MRCP, lab, or maybe your internal exam. These are the major GLUT transporters. If you look at the GLUT1 and GLUT3, they are found in most of the tissues. The most common thing, GLUT1 and 3 is found in most of the tissues, and the Km for them is 1 millimolar. So they have low Km value. Km is a Michaelis constant value. Low Km means the high affinity. High affinity means, let's say, if there is a low concentration of glucose in the blood, glucose is present in very, very low amount, still these transporters are going to uptake it. Affinity, Km, low Km, this is very important thing you should be knowing, means the high affinity. And how I remember it, affinity is very similar to the affection. So that particular transporter, GLUT1 and GLUT3, has a very high affection for the glucose. As soon as these transporters will see even a minute amount of glucose, they will uptake it inside the cell and then help in the digestion of that glucose molecules. Whereas the GLUT2 and GLUT4, they are found in the, GLUT2 is in the liver, GLUT4 is found in the skeletal muscles and the adipose tissues. But if you look at their Km value, they have very high Km value. It means whenever there is very, very high glucose concentration in the blood, it means you just had eaten a meal. Only then GLUT2 and GLUT4 transporters will be working. And where those are found? In the liver and the skeletal muscles and the adipose tissues. And if you go back and remind yourself, liver is the place where you store the glucose in the form of the glycogen. So whenever you have that makes sense also. Whenever you have a high molecule or high concentration of the glucose, you want to convert that glucose into the glycogen. Same is with the skeletal. Whenever you have the very, very high glucose level, then it can be stored in the form of the fats and lipids. That is, we can discuss in the lipid portion how we do that. Okay. So here is the normal fasting. It's hidden behind this is the fasting blood glucose in normal individuals is 10 to 100 milligram per deciliter, and the liver is the major organ that is involved in metabolizing and stabilizing the blood glucose level. So it is also known as the glucostat monitor. That is another key term or a question, MCQ question. What organ behaves as a glucostat monitor? What is the another name of glucostat monitor? It can be asked in various ways. So liver is a glucostat monitor. Okay, so metabolism of glucose, fructose, galactose, and mannose are key consideration, whereas out of all these four, glucose metabolism is the mostly asked one. Then the fructose is very important, so the galactose. The priority is for the glucose. And there are various major pathways, approximately 10, which are involved in the carbohydrate metabolism, namely glycolysis on the top, then the CAC, or you can say the Krebs cycle or the tricarboxylic acid cycle. CAC is the citric acid cycle, tricarboxylic acid cycle. This cycle has 3 names to it. Glycolysis has another name also, Embeden-Meyerhof pathway. I'll tell you again. Then gluconeogenesis, neogenesis. If you split this name, glucose, neo means new, genesis means synthesis. Newly synthesized glucose, okay, from non-carb precursors. Glycogenesis is the glycogen, genesis is the synthesis. Okay, in the biochemistry, whenever you hear a word, try to figure out the meaning. Like in Hindi, we used to do back, I don't know how many of you have taken Hindi as a subject. When in my school days, we used to have the Sandhi Vichhed. We used to break down the big complex term into smaller pieces. If you break these terms down, you will actually get the meaning behind the names. Glycogenesis. Glycogen synthesis is glycogenesis. Glycolysis. Breakdown of glycogen is glycogenolysis. Hexose monophosphate shunt, uronic acid pathway, galactose metabolism, fructose metabolism, amino sugar, and mucopolysaccharide metabolism. These are the major pathways which should be studied under carbohydrate metabolism. If you look at all of them quickly, one by one, the summary of all of the pathways, glycolysis, also known as Embeden-Meyerhof pathway. In this, glucose is oxidized to pyruvate or the lactate. Pyruvate is in the presence of the oxygen, plus oxygen means in the presence of the oxygen. In the absence of oxygen, it is converted into lactate, and that we will see in today's lecture. Second is the citric acid cycle, Krebs cycle, or TCA cycle. In this, acetyl coenzyme is oxidized. This is an oxidation. Oxidized to carbon dioxide plus the ATPs, which is not seen here. Okay. Final common oxidative pathways for the carbs, fats, amino acid. I have told you that like it is similar that all the rivers finally merge into the ocean. So all the 3 of the metabolic pathway, be it your protein, be it your fat, be it your carb, they finally merge into the TCA cycle. Gluconeogenesis, in this, the known carb precursors is from the known carb precursors, glucose is synthesized. And when you will synthesize the glucose, when you don't have the glucose in your diet or you're not eating, you are in the starvation phase. Okay. Glycogenesis, when there is a plenty of glucose, you want to store the glucose so that you can use it later, like you store everything in a storehouse or in the closet in your home. You store the glucose in the form of the glycogen inside the cell. And whenever you need the energy in the starvation state, you want to open the energy store and take the glycogen out, chop it off into the glucose and consume that glucose to get the energy required by the body. That is glycogenolysis, which is glycogen breakdown into the glucose. Okay. And other are the hexose monophosphate shunt, in the short known as HMP, or another name is PPP, pentose phosphate pathway. It is also known as the direct oxidative pathway. Names are very important for examination point of view. I always emphasize my student to learn also known as. You should be knowing it, because just to mislead you, they can use the term the direct oxidative pathway. Now, when you are reading the question, you know the PPP pathway, but you don't know what the hell is this direct oxidative pathway. Okay, so PPP, HMP, and the direct oxidative pathway is the name of a similar pathway. This is the alternate of the glycolysis and the TCA for the glucose oxidation. Okay, uronic acid pathway, in this glucose is converted into glucouronic acid, that is a pentose. Glucose is a hexose. Hexose means it has a 6 carbon story, whereas glucouronic acid is a pentose, 1 carbon less. Okay, galactose metabolism. Galactose is converted to glucose. If you go and check in the stereochemistry, they're apimers of each other, actually, galactose and glucose. So this is what galactose metabolism is. You have an epimerase, you can flip the structure, and then that is converted into glucose, and it is used as the similar regular glucose that you have in the stock. Okay. Fructose metabolism, in fructose is metabolized to pyruvate, and then after the pyruvate, it is taken down to the common pathway, that is the TCA cycle, and used in the same regular way. The amino acid sugars and the other sugars are used for the formation of mucopolysaccharides and glycoproteins, which are part of your cell structure. It is a very, very important part of a cell structure, mucopolysaccharides, glycoproteins, right? So in this, I was just trying to show you that glucose has 2 kinds of a pathway, either the oxidative pathway or the synthetic pathway. Your glycolysis, HMP, and uronic acid is oxidative pathway. This is important. This is important. Oxidative pathways are glycolysis, HMP shunt, and uronic acid pathway. There is a similar question in the exam about this anaplerotic reactions. So this term is really used with those pathways which has both the phases to it, which has both anabolic process, means synthesis is going on, and the catabolic process. It means the breakdown is going on. And there are only 2 pathways which you will study in the carbohydrate metabolism, that is citric acid cycle and the glycolysis, which has both the anabolic and the catabolic pathways associated to it. Or they both are also known as amphibolic pathways. Remember in +1, +2 classes, you used to use words, the amphoteric, which has both the acidic and the basic properties. Similarly, when a pathway has both the catabolic, the breakdown process, anabolic, the synthetic process going on, that pathway is the amphibolic pathway, and the citric acid cycle and glycolysis both are the examples of the amphibolic pathways. Okay. Now, whenever you will study the metabolic pathways, so always make a checklist that you should be knowing about the pathway, about like these things you should be knowing about. Organ, where it is occurring, compartment, which compartment is involved. It is occurring in the cytoplasm or the mitochondria or the lysosome or Golgi apparatus, where this is going on. What are the substrates to it? What are the donors which are giving maybe the phosphate group? Maybe the electron is coming. Okay. What are the end products? Most important is the rate limiting step. Rate limiting step is the slowest step. Like you can imagine you are in a group of 10 and all of you have to run to win the race. So 10 should run at the highest speed. So if 1 is the slowest person out of 10, so your entire speed will be dependent upon that slowest person, because unless that slowest damn fool will not run fast, you are not going to win. Okay. So the rate limiting pathway, rate limiting step is the slowest step that limits the entire reaction pathway. Okay. So we have to usually push it up, and you should be knowing that what is the rate limiting step. Enzymes, which are targeted for regulation. Remember guys, now is the stage when you should be knowing the regulation most, because body has to maintain the homeostasis no matter which is thing you are studying. Whenever you are talking about a disease, we say nowadays also your immunity, boost up your immunity, Sometimes you have the vitamins to take for to fight for an infection, sometimes this, sometimes that. Every time what we are doing is we are taking some kind of like chemicals or compounds, maybe an allopathic medicine, maybe an herbal medicine, maybe a home remedy, so that your body get back to its homeostasis, right? Being you as a doctor, you diagnose the disease, but the ultimate aim is to put back the body into its original place. And that original place is disturbed because the regulation is disturbed. So always and always and always, whenever you learn anything or study any topic, you should have like a Arjun Nishan, right? You should have a bullseye. That is what is the key regulatory enzyme, key regulatory step. Because if that part is disturbed, then disease is going to happen. The entire mechanism is going to get disturbed. That's what I want to say. Energetics, how much energy is involved. Okay, so our major focus is glycolysis, which should be done in, I guess, in 10-15 minutes. Then I have something to discuss. Glycolysis, it occurs in almost all cells, right? It is a cytoplasmic pathway. It occurs in the cytoplasm, in which one molecule of glucose is converted into 2 molecules of the pyruvate. Okay, and energy captured in 2 substrate level phosphorylation. There are 2 SLPs involved, substrate level phosphorylation, and there is 1 oxidation reaction. What do we mean by that? Those who have studied the enzymes, they can figure out the oxidation reaction. Whenever there is an oxidation reduction reaction, you will see 1 NADPH is involved. You will definitely see 1 NADPH. Either it will be, let me make it both ways. Okay, it can go both ways. If one is reducing, this is oxidation. So oxidation reduction, they happen continuously. So there is 1 NADH involvement that we will see. Now, what is substrate level phosphorylation? See, there is an oxidative phosphorylation. Oxidative phosphorylation is the one when we are talking about the getting the energy, when we are talking about formation of ATP, right? Formation of ATP via electron transport chain. But in the substrate level in oxidative phosphate, you have the oxygen involved. In the substrate level phosphorylation, you don't have the oxygen involved. Still, you are synthesizing ATP. ATP can be synthesized. Okay, that is substrate level phosphorylation. That is very important. It occurs aerobically and anaerobically. It is very glycolysis in cardiac muscle is low. This is a very, very important, I will say. The cardiac muscles have the least tendency to undergo glycolysis. So you can imagine from where the cardiac muscles are deriving its energy from. Cardiac muscles are deriving its energy from where if glycolysis is not happening there. Go home and check. You will find very amazing answer to it. Okay, so now I will stop this and I will start you how we take the class. Actually, the class, this is my whiteboard. And today we will be discussing about the glycolysis. Okay, that is conversion of the glucose to the pyruvate or maybe the lactate, the various steps that are involved, various enzymes that are involved, and how those enzymes are regulated and what are the disease associated with it, and how which inhibitors can block which step. So all these things I'll discuss. Starting it with the glucose. With the glucose. You have the glucose that is transported by maybe GLUT1, GLUT2, GLUT3, GLUT4. Now you have the glucose. Right? Now you have this glucose is inside the cytoplasm, which will get converted into glucose 6-phosphate. Name is saying what? You have one phosphate. And that phosphate always and always, just make it a routine, will come at the expense of the ATP. So ATP means you spended 1 rupee, or you can say 1 note or $1. So you have spended 1 ATP to make the glucose 6 phosphate, and that is done by the enzyme hexokinase. Now there is one funny thing actually here. Sometimes you will see the hexokinase, sometimes you will see the glucokinase. And many times, many students come to me, what is the difference, ma'am, between the hexokinase and the glucokinase? Remember, both the enzymes are magnesium-dependent enzyme. Every enzyme has a coenzyme or a cofactor related to it, which helps to maintain its activity. Okay, they can be tightly bound, or maybe whenever it is needed, they can be activated. So glucokinase and hexokinase is magnesium-dependent enzymes, and that convert glucose into glucose 6-phosphate at the expense of 1 ATP, and that is an irreversible step. What is the difference between glucose kinase and the hexokinase? Here, you can make this table. Glucokinase, and this is hexokinase. Just make a table. Okay, what is the difference? Glucokinase is found only in, liver and pancreas. And hexokinase is active in all tissues. It is there in all tissues. Right? And this has a low affinity. Low affinity, or you can say high Km value. And this has the high affinity, or you can say the low Km value. It is inducible. And this is inhibited by glucose 6-phosphate. I'm sorry, the light has gone, so probably you will see my funny face, but the internet will go on. Okay. So these are the difference between the glucokinase and the hexokinase enzyme. So if you can see, they are present in all the tissues. What it is making them important is they are present in the all tissues. It means they should pick up the glucose whenever there is a glucose available. So it means they should have a high affinity whenever there is even low concentration of glucose is there. Okay. Now let's come back to this. Glucose is converted into glucose 6-phosphate, which can be converted into fructose 6-phosphate by enzyme PFK, my favorite enzyme, PFK1. PFK1 stands for phosphofructokinase. All the way here in Chandigarh. So that's why this light issue is going on. Though I have arranged the backup for the internet, but definitely not for the light. Something failed. So PFK1 is the phosphofructokinase. It has 2 versions to it, PFK1 and PFK2. It converts glucose 6-phosphate into fructose 6-phosphate enzyme. Now, fructose 6-phosphate can be converted into fructose 1,6-bisphosphate. Bisphosphate. Bis means 2. Sometimes for 2 we use as a di, bi, and bis. It's all the same thing. 2 things to it. It means bisphosphate. 2 phosphates are there. And it means we are introducing 1 phosphate, and that 1 phosphate is coming at then again the expense of ATP. Okay, so now we have consumed 2 ATPs so far, one for the formation of the glucose 6-phosphate, then here for the synthesis of the glucose 6-phosphate, and another for the synthesis of the fructose 1,6-bisphosphate. So how many steps we have studied so far? 1. This is the first step, this is the second step, and this is the third step. These 3 steps, I call them as the preparatory phase or energy expenditure phase. Like the one, let's say you are in a new job. So initially you have an induction phase where you will be taught at the expense of the company or the facility or maybe the hospital. They will spend energy, time, and money on you so that you get well equipped with how the entire thing is going on, the process is going on in the hospital. So this steps is the preparatory phase. And the steps after it, all those are the energy yielding phases. You will get the energy in those steps. So fructose 1,6-phosphate, if you clearly see, it is the 6 carbon thing. 1, 2, 3, 4, 5, 6 carbon thing. And that if we chop it off into 2 pieces, that gives almost exactly looking 2 entities, DHAP and G3P, I call it. DHAP is the dihydroxyacetone phosphate and G3P is the glyceraldehyde 3-phosphate. They both are the isomers of each other, and this is done by the enzyme aldolase. Right? So aldolase chops it off. The aldolase nature is to snip it off like a scissor. So it chops off the fructose 1,6-bisphosphate into 2 entities, DHAP and the glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Now, this is another very important step here, is get converted into 1,3-bisphosphoglycerate. Now the name is suggesting there is 1,3-bisphospho. 2 phosphate groups are there, right? So it means something has added to it, which added 1 phosphate to it. Is it that expense of ATP? No, this is not at the expense of ATP. Rather, there is this entity, we call them inorganic phosphate, PI. It is indicated by PI. At the expense of the inorganic phosphate, you get 1,3-bisphosphoglycerate. This is a high energy compound, which gives off the energy. So from low energy, this is the low energy thing. This is a high energy thing. Now, when you will make changes in the certain changes in the structure or try to snip off the inorganic phosphate, it will give you the ATP. Right? And the enzyme here, after this, you will get 3-PG, 3-phosphoglycerate. From 3-phosphoglycerate, you make 2-phosphoglycerate. And then from 2-phosphoglycerate, you make phosphoenol pyruvate. Why I'm jumping? This is important. Phosphoenol pyruvate is again the high energy compound, which ultimately gives you the pyruvate, which we were looking for. And the enzyme involved here is the pyruvate kinase. This enzyme is again very important regulatory enzyme in this, and it is associated with the hemolytic anemia also. There is this glyceraldehyde 3-phosphodehydrogenase deficiency, that is inherited deficiency. That is the major cause of hemolytic anemia in the patient. And the second one is the pyruvate kinase deficiency. The difference between this G3PD deficiency and the pyruvate kinase deficiency is Heinz bodies are there involved. As whenever you have a case study, you will see the patient, from the patient testing, the doctor has figured out the Heinz bodies. You will see the ketone Heinz body. That is indicative of glyceraldehyde 3-phosphate dehydrogenase deficiency. Whereas the pyruvate kinase deficiency causes the hemolytic anemia without the Heinz body present. So this is the key difference between these two deficiency disease, and this is one of the regulatory enzyme also, right? So here I have told you 3 important enzyme to know. Glucokinase or hexokinase is important enzyme. Then phosphofructokinase 1 is important enzyme, and the pyruvate kinase. These are the 3 very important enzymes that are related to this glycolysis. And if we look at the phosphofructokinase 1, let me tell you in detail about phosphofructokinase PFK1. PFK1 is doing what? It is converting fructose 1-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate. So what happens in the body is few very minute amount of this fructose 6-phosphate is converted into fructose 2,6-bisphosphate. This is 2, this is 1. This is a very, very minute quantity that is converted by PFK2 enzyme that is present, very minute quantity. And this product actually activates phosphofructokinase 1. So this is a very important regulatory step. Who is regulating PFK1? PFK1 is regulated by fructose 2,6-bisphosphate, which is a product of fructose 6-phosphate itself, but that is converted by phosphofructose kinase 2 enzyme, and that regulates this. And whenever there is low ATP, or you can see there is a low energy, it means there are high levels of AMP. AMP is a low energy indicator. Then it will trigger the phosphofructokinase to work very fast. Similarly, there is citrate. Citrate will help the process to speed up. And if we talk about, let's say, the hormones, the majorly we discussed hormones are your glucagon. Glucagon is there when there is low level of glucose. Insulin presence means there is a high level of glucose. So insulin has a positive effect for this reaction. Glucagon has a negative effect. Insulin means high glucose. That makes sense also. When there is a glucose, you want to break down the glucose to get the energy, right? And whenever there is a glucagon, it means there is a less amount of glucose. When there is a less amount of glucose, that is going to inhibit the process to save the energy or slow down the process. So this is kind of a homeostasis mechanism your body has. Second thing I want to discuss here is 1,3-bisphosphoglycerate. Very, very important thing. There is a similar 2,3-BPG in the short form. 2,3-bisphosphoglycerate. In the erythrocyte, you have bisphosphoglycerate mutase. Mutase is kind of an epimerase which converts this into back and forth. There is a kind of mutase in the erythrocyte. So whenever you have the low oxygen levels, 1,3-BPG is get converted into 2,3-BPG. And what the 2,3-BPG has? This 2,3-BPG lowers the affinity of hemoglobin for oxygen. So you can imagine on the higher altitude, whenever there is a low concentration of oxygen in the atmosphere, the erythrocytes convert 1,3-BPG into 2,3-BPG. So the oxygen dissociation curve, which exactly looks like this is the oxygen dissociation curve, will shift towards the right. This is 2,3-BPG. This will help the hemoglobin to unload the oxygen very quickly to the tissue so that they can survive. Now, one important thing here is fetal hemoglobin. Fetal hemoglobin has very, very low affinity, almost negligible. It doesn't like 2,3-BPG. It has very high affinity for 1,3-BPG. That's how the mother's hemoglobin oxygen get transferred to the fetal hemoglobin when the baby is in the womb, right? So that shifts the oxygen curves towards the fetal hemoglobin, shifts the oxygen curves towards the left. This is the fetal. So this is this compensatory mechanism of 1,3-BPG and 2,3-BPG in your body. Okay, so you have, if you look at the total energetics here, so you consume 2 ATPs, one is right there, another is right there. And you get 1 ATP there and 1 ATP here. In this reaction, and there is one NADH that is involved, that is this NAD positive give you NADH. So 2 energy ATPs are consumed, 2 ATPs are released, and 1 NADH is released. Is that true? This is times 2, because from 1 fructose 1,6-bisphosphate, you have 2 glyceraldehyde molecules, and DHAP and glyceraldehyde 3-phosphate, they are isomers of each other. So DHAP finally gets converted into G3P by isomerase enzyme. So everything after this is times 2. So in total, you have 4 ATP here. NADH is 1.5 ATP or 2.5 ATP. That depends upon which shuttle it takes. So let's say it is 1.5 ATP, right? So it is almost 3 ATPs. So 4 plus 3 is 7. Times 2 is 2 NADH. 2 NADH times 2.5 ATP gives you 5 ATP. So total of 9 ATP you have produced and minus 2 ATPs you have consumed. So nutshell of 7 ATP production for 1 glucose molecule up to the pyruvate. So that is the net energetics. Yes, but we have various other inhibitors. We are short of time. I have to wrap this up. So I cannot go, but there are several other energetics metabolism which changes the total energetics of, sometimes you have the 5 ATP, sometimes you have the 7, sometimes you have the 10, sometimes you have the 6 ATP. That is coming from the glycolysis cycle that we can discuss some other time. But in the end, I want to give you this. In next 5 minutes, I should wrap up. This is a pseudo glucose, not true glucose, but this 2DG that is coming out in the market in a couple of days from DRDO. That is a new oral drug for COVID-19. And this pseudo glucose means this is a mimic. This is mimicking the glucose. This is not exactly the glucose. It is named 2-deoxyglucose. 1, 2, 3, 4, 5, 6, right? And all these 6 carbons, they have the hydroxyl group attached to it. I'm not doing the left and right here. in 2-deoxyglucose, the second number carbon, hydroxyl group is getting replaced with the hydrogen. So that's the name 2-deoxyglucose. So we are fooling a person or a COVID virus, which is actually fooling our mechanism, right? So this is a mimicry. The COVID virus is SARS-CoV-2 virus is entering into our system, utilizes our system, and finally infecting us. So that is bad. So what 2-deoxyglucose is going to do is it is going in the viral system as a glucose, mimicking the glucose and thereby killing it, because the virus needs the energy in the form of a glucose. But glucose is not there. 2-deoxyglucose is there, which is not going to give the ATP. Ultimately, it will die in next 10 days, right? So it is fun, actually. This is done by Dr. Anil Kumar, and he is a scientist, actually senior scientist with this DRDO, and that has given us this, and government has given the approval. It is out there. It should be out there in the market in the couple of the days. This is known as the miracle worker, and the phase 2 clinical studies have been done, right? And finally, the government has released it in with a very good company also. So what it does is it reduces the dependence on the supplemental oxygen also, right? What does is it deprives the COVID from the food, and without the food, the virus is dying in 10 days, because that is almost the life cycle we talk about for this SARS-CoV-2 virus. Extensive clinical trials have done, and those are done in India. So it means it is done on us in our genetics, not in somebody else's ethnicity. So we are of Asian, we are Indian, so it works with our ethnicity. That is done good. Phase 1 trial was good. Phase 2 was for 110 patients, and all of these patients are very seriously. They are not the mild and the moderate one, but the serious patients they have included in the clinical trials, right? And these are phase 3 clinical trials was done on 220 patients, and the results were it is the safe. There are no side effects that has been observed, and it is showed the faster symptomatic cure, and same results were there in all age of the patients, whether they are the young or the old. So that is the good news, right? So advantages is here is it's a generic molecule, and it is an analog of glucose. I have told you that, right? And 27th May, first batch will come in the market, and I have this from Dr. Reddy's actually it is coming. This is a packet, and this is developed in the collaboration with the DRDO. And that is a good news for all of you and for me also. So be safe, stay home, and whenever there will be chance, we will connect to you again. If you have any questions or any concerns, you can ping me, you can write to me. I'm writing my email ID here, vgusp2013@gmail.com. That is my personal ID. You can connect to me, or you can connect to your coordinators. They will get back to me. If you have any questions, please let me take the questions. Otherwise, I'll keep on talking. Thank you very much for listening. See, I had the same question in my mind, because in 2019, this drug 2DG was approved for treating the cancer. But the studies have said that cannot be used alone, but it should be done in combination with other drug. So that was I'm talking about for the cancer treatment. Then from there, that again question came in the mind, and I was trying to look at it whether it is given in combination with something or not. And to my surprise, though I couldn't found any paper, I can share you the things also. I did a lot of research. If I share you my screen, but they are really not saying anything that it is having any effect actually, have any effect actually on deprivation of other normal cells. I have went through everything actually here. If I close this, okay, this is the data I was going through. Data suggests for the COVID drugs, and they are not saying anywhere that it is going to do that. And I couldn't find any paper. Maybe I'm doing more research on it, but can't say anything. Maybe I will come back to you on this sometime later if I can find out something, though I have written to Dr. Anil to give me more insight on this. Yeah. Any other question? I cannot hear the question. Um, Actually, Kaplan is my favorite, to tell you the truth. But here, if I have a book from the DBMCI, actually, if you want to get this book also, that is a Biochemistry Complete, that gives you, actually, I have concised the tables and the key terms that you should be knowing for the exams. But specifically for USMLE, you should be able to understand every single word from the Kaplan. Plus, you should be having the question bank. This is the Kaplan book, actually. Even I referred this book. And then you have these Q banks online. UWorld is the best, which everybody is using, though even we have a hell lot of questions. I myself made maybe 600 plus questions for the biochemistry. If you practice those questions, and if you study to my case studies, I prefer my case studies, because being a researcher and being the new in this new era, we should be going on the research based um, uh, diagnosis for every patient. So that should suffice the purpose. The Kaplan and the UWorld question banks, then the question bank from us, plus this Biochemistry Complete by DBMCI. That is all actually. Next question, please. I will say start studying the chapter on enzymes first, then do every chapter one by one. Just read it. When you will do third time, when you will read the entire thing third time, then you will start correlating the things. See, biochemistry is interconnected with everything, like the nerves inside your body. So more you study the same topic over and over, every time you will see you found out one thing. So strategy is keep it very simple. I have a very much like videos of the mnemonics. So cycles, learning cycles and tables, they are very easy to tell you the truth. I'm saying it because I'm confident about it. So more you will listen to me, my lectures, more you will go through the studies, case studies given by me and the question banks, you will get to it. That is my promise to you. See, Vasudevan is good book. As an Indian book, I have looked upon the Satyanarayana also, but they are very limited books. But if you go for the Lippincott, if you go for the other molecular biology or the medicinal chemistry, see, Lippincott has a portion which is good for nucleic acid, but it is not good for pathways. So every book has a positive and a negative to it. So it is always, this is the same question I get to hear from everybody over and over. So I'm not doing the propaganda of myself, but being me as a teacher, it can be any other teacher. Teacher reads all those 4 or 5 books, summarize it, and gives you the entire thing. So you can pick any book. Maybe you can start with a Harper, maybe you can start with the Lippincott, but mug it up. Don't jump from books to books. I personally prefer international author books because they look very vast, but when you start reading it thoroughly, you get an entire big picture because you cannot cram everything. When I was a student in India, I'm born here, brought up here, but since almost 20 years, like 2 decades, I'm there in US. The difference I have found out is being us and an Indian would try to muck things up. Okay, let's cram this, ye yaad kar liya, wo yaad kar liya. But that is not going to work now. You have to understand, because what we understand, we retain it better rather than learning it. Don't try to memorize as such, mug it up, ghot ke pee liya. Try to understand. Understanding will come from the foreign author book. You pick the Lippincott if you have one. You pick the Harper if you have one, but read it thoroughly. Stick to it thoroughly. Yeah, let me share you something here. The same thing I will share. Yeah. Say, I know, I do cannot. Let me stop it. Okay, let me stop it and let me verbally say it, read it out from there. So you have this spike, glucose thing in there, in this, that DG targets. Okay, so this protein is the target for the SARS-CoV-2 virus. It targets it. Okay, and 2DG targets that SARS-CoV-2 via this spike protein. It's a little bit complicated. Simplest way in this short period of time is, as soon as it gets into the cell, SARS-CoV-2 virus is extracting the energy from the cell. It is like, I myself see it not as a virus, but as a parasite. It is sucking up in our cells. So we, rather than giving them the healthy food, we give them this false food in the form of the 2DG. So glucose is going to give them the ATP, right? The SARS-CoV-2 as an ATP. So it is not getting ATP now. It is sucking up. If you go and look at the glycolysis, it 2DG gets converted into 2DGP, right? Glucose is getting converted into glucose 6-phosphate. Now you don't have glucose, you have 2DG. That 2DG gets converted into 2DG phosphate. Now the hexokinase and the glucokinase is stuck. So entire glycolysis pathway is fallen apart. So whenever ATP is needed for the SARS-CoV-2 to multiply, it is not there. It is going to die eventually. That is the simplest way I can tell it right now. Okay.
Thank you, ma'am. That was yet another informative session. I'm sure that the session has succeeded in strengthening the basics in biochemistry. On behalf of IMA MSN family, I would like to thank Varsha, ma'am, who handled the session, and all the participants who made this event successful. Never stop learning because life never stops teaching. Thank you.
Thank you for having me. Have a good day. Bye. Take care and be safe.
Thank you, ma'am.
Thank you.